हेलो डियर स्टूडेंट्स, टारगेट 90 प्लस की सीरीज़ में आप सभी का स्वागत है। आज हम देखेंगे चैप्टर नंबर 4 मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस। लेकिन ये स्टार्ट करने से पहले चैनल को लाइक, सब्सक्राइब एंड कमेंट करें। बोर्ड एग्ज़ाम डेट से पहले आपको सारे चैप्टर्स के क्वेश्चन आंसर्स मिल जाएंगे, तो प्लीज़ लाइक करें और शेयर करें। इसका वेटेज है 4 to 6 मार्क्स। तो चलिए स्टार्ट करते हैं क्वेश्चन आंसर्स सेक्शन 1, जिसमें 1 मार्क क्वेश्चंस हैं। व्हाट इज़ द प्रिंसिपल ऑफ डीएनए प्रोफाइलिंग? इट इज़ बेस्ड ऑन आइडेंटिफिकेशन ऑफ न्यूक्लियोटाइड सीक्वेंस प्रेजेंट इन डीएनए मॉलिक्यूल। व्हाट इज़ द यूज ऑफ सदर्न ब्लॉटिंग इन डीएनए फिंगरप्रिंटिंग? सदर्न ब्लॉटिंग जिसमें डीएनए फ्रैगमेंट्स आर ट्रांसफर्ड ऑन द नायलॉन मेम्ब्रेन। एनलिस्ट द जीन्स प्रेजेंट इन लैक ओपेरॉन। लैक ओपेरॉन के आपको स्ट्रक्चरल जीन्स के नाम लिखने हैं, जिसमें लैक जेड, लैक वाई एंड लैक ए हैं। व्हाट इज मेंट बाय ओपेरॉन? इट इज अ ट्रांसक्रिप्शनल कंट्रोल मैकेनिज्म ऑफ जीन रेगुलेशन। नेम अमाइनो एसिड इन प्रोकैरियोट्स एंड यूकैरियोट्स कोडेड बाय AUG कोडॉन। AUG ये स्टार्ट कोडॉन है और ये अलग अलग अमाइनो एसिड कोड करता है प्रोकैरियोट्स एंड यूकैरियोट्स में, जिसमें methionine in eukaryotes and formylmethionine in prokaryotes. What is meant by activation of amino acid? When amino acid is bound with tRNA with the help of ATP, it is called as activation of amino acid. What is the role of Mg plus in translation? ये Mg कोफैक्टर का काम करता है जो स्मॉल एंड लार्ज राइबोसोमल सबयूनिट को बाइंड करता है। What are different types of mutation? 2 types, point mutation and frame shift mutation. Enlist the enzymes used in semi-conservative DNA replication. आप कोई भी 2-3 लिख सकते हैं। हेलिकेज, DNA polymerase, DNA ligase, DNA gyrase, topoisomerase. What is central dogma of molecular biology? It is unidirectional flow of information from DNA to RNA to proteins. What type of isotope used in semi-conservative DNA replication experiment? It is N15. What is the function of RNA primer? It provides free end for the addition of nucleotides during the DNA replication. Give function of SSBPs. SSBP, the separated strands of DNA are prevented from the recoiling by SSBP. SSBP is single strand binding proteins. यहाँ पे P extras हो गए हैं. Define RFLP, restriction fragment length polymorphism. When restriction enzyme cut DNA into small fragments having variable length, this is called RFLP. Define heterochromatin. It is highly condensed region of chromosome which is genetically inactive, and it is located near centromere and telomeres. What is anticodon? It is the part of tRNA that are complementary to codons present on mRNA. Enlist the histone that forms octamer of nucleosome, 2 copies of each, H2A, H2B, H3 and H4. Define genome. It is the total genetic constitution of an organism. Give full form of HGP, Human Genome Project. Give full form of VNTR, Variable Number of Tandem Repeats. Why genetic code is commaless? Because there is no gap or punctuation mark between successive codon, therefore it is commaless. What is degeneracy of genetic code? Single amino acid is encoded by single codon, but when one amino acids are encoded by more than one codon, this is called degeneracy. Now 2-3 marks question. Discuss any 2 points due to which Mendel got success in his experiment. यहाँ पे 4 मेंशन किया, आप कोई भी 2 लिख सकते हैं. He planned his experiments carefully and involved large sample size. He recorded the number of plants of each type, and expressed his results in ratio. The contrasting characters can be easily recognized. He introduced the concept of dominance and recessiveness. Differentiate between heterochromatin and euchromatin. Heterochromatin, it is highly condensed region. Euchromatin, it is non-condensed region. Because highly condensed, it's chain dark, it's chain light. They are genetically less active, they are genetically more active. They are located near centromere and telomere. They are located in the middle of the chromosomes. How tRNA act as adapter molecule? The 3' end of tRNA has CCA sequence that accept amino acid with the help of ATP. For every amino acid, there is specific tRNA. The tRNA has ability to read the codon and bind to the amino acid. Therefore, it is called adapter molecule. Define mutation and give its two types. It is sudden change in the sequence of DNA that result in changed phenotype. There are two types, point mutation and frame shift mutation. Describe Hershey and Chase experiment. So the diagram is given here. कभी कभी सिर्फ डायग्राम पे क्वेश्चन आता है और कभी पूरा एक्सप्लेनेशन के लिए आता है. So in Hershey and Chase, they used bacteriophage and E. coli, that is virus. They grew bacteriophage in radioactive P32, P33 and S35 medium. The viruses that grown in P33 shows radioactive DNA, and the viruses that are grown in S35 showed radioactive protein. These radioactive phages were allowed to infect E. coli cells. After the infection proceeds, the viral coats were removed from the host by centrifugation or blending. The radioactivity measured in the cell and supernatant. The bacteria which were infected by the virus having radioactive DNA were showing radioactivity. This concluded that DNA entered into the host cell and act as genetic material and not the protein. Explain the role of inducer in lactoperon. When lactose enters in the cytoplasm of the cell, it is converted into allolactose. This allolactose binds to the repressor protein and make operator gene free. This allows the binding of RNA polymerase and begin the process of transcription. Draw label diagram of nucleosome. So here you can see there are 8 histone proteins, H2A, H2B, H3 and H4. There are 2 copies of each, and H1 green colored linker histone and red colored DNA. Write a note on packaging of DNA in prokaryotes. The prokaryotes do not have well organized nucleus and there is no nuclear membrane. Their nucleus is called nucleolus. The size of prokaryotic DNA is approx 1100 micron long. It is circularized so that diameter is reduced to 350 micrometer. It is further reduced to 30 micrometer by folding into 40 to 50 loops. These loops are connected by RNA connectors. Each loop is further coiled and supercoiled and reduced the size up to 2 micrometer in diameter. And it is further assisted by enzyme like DNA gyrase and topoisomerase. The diagram is also given here. So पहले circular DNA है, then folding हो रहा है and supercoiling. Write a note on packaging of DNA in eukaryotes. So eukaryotes have well organized nucleus. They contain nuclear membrane, nucleolus and chromosome. The approx length of human chromosome is 2.2 meter. These chromosomes are packed with the help of histone and non-histone proteins. Histones are positively charged proteins rich in amino acids like lysine and arginine. This is very very important. इस पे 1 mark question separately आ सकता है. Name the amino acids present on histone, lysine and arginine. The negatively charged DNA coil around the histone octamer and form nucleosome. The H1 histone protein bind to DNA at entry and exit. The length of DNA in one nucleosome is 200 base pair. The repeating nucleosome look like beads on string. 6 nucleosome coil to form solenoid structure, and it is further supercoiled to form a chromatin fiber to form chromosome structure. Draw neat label diagram of replication fork. So you can make this diagram: leading, lagging strand, primers, template and replication fork. Explain every McCarthy and MacLeod experiment to prove DNA as genetic material. They obtained cell-free extract of estrine by heat killing and centrifugation. They treated the cell-free extract with different enzymes like RNase, DNase, protease. The cell-free extract treated with enzymes are mixed with live R bacteria. They found that protease and RNase did not affect the transformation, whereas DNase affected the transformation. This proves that DNA is a genetic material or transforming principle. What is the goal of Human Genome Project? Mapping the entire human genome at the level of nucleotide sequence to store the information collected from the project in database form, to develop tools and techniques for analysis of this data, to transfer the related technology to private sector, and taking care of legal, ethical and social issues. Which materials can be used to isolate DNA in fingerprint technique? Give its application. So any cell or tissue like blood, root hair cells, etc. Applications of fingerprinting in forensic science: to solve problems of rape and murder cases. It is used to find out the biological father or mother of child. It is used in pedigree analysis of animals. Explain Griffith experiment with diagram. So Griffith used Streptococcus pneumoniae, and there are two strains. S stand for smooth, capsulated and virulent. R stand for rough, non-capsulated and virulent, means non-pathogenic. So he conducted 4 experiments. In his first experiment, he injected live R into mice and mice survived. In second experiment, he used live S strain injected into mice and found that mice died. In third experiment, he used heat killed S strain and found that mice survived. In his fourth experiment, he used heat killed S strain and live R strain injected into the mouse and find that mice died because of pneumonia. He observed the blood of died mice and found that S bacteria were present. So he concluded that R bacteria must have taken up something from the heat killed S, due to which R strain got transformed into S and mice died. Describe the characteristic of genetic code. It is triplet code, consists of 3 nucleotides. One genetic code specifies one amino acid. It is non-overlapping, commaless. It has distinct polarity 5' to 3'. It has degeneracy, means some amino acid have more codon. Genetic code is non-ambiguous, that is, specific amino acid is coded by particular codon. There are initiation codon and termination codon. Initiation codon is AUG and termination codons are UAA, UAG, UGA. There are 3. They are also called as stop codons. Draw a label diagram of Michelson and Stahl experiment and also explain it. They used equilibrium density gradient centrifugation technique. They cultured E. coli bacteria in heavy isotope N15 medium for several generations. The DNA was extracted and centrifuged in cesium chloride gradient. These E. coli cells were later transformed into N14 medium, and after first generation, the DNA was extracted and centrifuged. After second generation, the DNA was extracted and centrifuged, and the band positions were noted. The E. coli grown in N15 medium showed band position at the bottom. After first generation, the band position was intermediate, and after second generation, it showed that intermediate position and light bands in upper layers. The position of bands after two generations clearly indicate that DNA replication occurred by a semi-conservative method. The diagram is given here. Explain the steps of DNA fingerprinting. First, isolation of DNA. It can be done from any cells like blood, hair, roots, skin. Then second step, restriction digestion. All the isolated DNA is digested with the use of restriction enzymes, and DNA fragments are produced having different length called RFLP. Third step, gel electrophoresis. The fragmented DNA is electrophoresed on agarose gel, and here it is separated based on charge and length. Southern blotting. The double stranded DNA from the gel are transferred to the nitrocellulose paper. Selection of DNA probe. The known sequence of DNA radio labeled. Hybridization. The probe is added to nitrocellulose filter paper and it binds to the complementary sequence. And last is photography, where the nitrocellulose filter paper is photographed on X-ray film. Explain the concept of operon. It is a transcription control mechanism of gene regulation. The operon consists of repressor gene or regulatory gene. It produces repressor protein that bind to the operator gene. It also has promoter gene, which is the site for RNA polymerase binding. Operator gene, it is the site for binding of repressor protein. Structural gene, example, lac operon has three structural gene that produces three different enzymes. Lac Z, lac Y, lac A produces beta galactosidase, beta galactosidase permease and transacetylase. Give diagrammatic representation of lac operon in presence of inducer. So here the diagram is given. Define genomics and give its application. The total genetic constitution of organism is called genome. The study of genome with its analysis, sequencing and mapping with those function is called as genomics. Applications. Improvement of crop and human health. It is used in medicine and biotechnology. It is used in treatment of genetic disorders through gene therapy. It is used to introduce new genes in the microbes for the production of enzymes, proteins, microbes and biofuels. Describe the process of semi-conservative DNA replication with the help of diagram. So activation of nucleotide, first step, in which nucleotides are phosphorylated. Point of origin, prokaryotes have only one origin site, whereas eukaryotes have many. Unwinding of DNA. So double stranded is converted into single stranded by breaking hydrogen bond using helicase enzyme. The single stranded binding protein prevent the recoiling, and the point formed is called replication fork. Each strand acts as template and RNA primers attach at the 3' end of the template. The DNA polymerase forms new strand in 5' to 3' direction by adding nucleotides. Leading and lagging strand. The new strand is always formed in 5 to 3 direction. So both strands are anti-parallel. One develop continuously towards the fork called leading strand and other develop away from the fork called Okazaki fragments. And later these fragments are joined by DNA ligase. Describe translation with the diagram. So translation is converting mRNA into protein. So there are three steps: initiation, elongation and termination. In initiation, the amino acids are activated. The amino acids are bound to tRNA and form amino acid tRNA. The first tRNA containing methionine binds to the start codon. This allows the binding of small and large subunit of ribosome with the help of Mg plus cofactor. The ribosomal complex forms three sites E site, P site, and A site. Elongation. The initiated tRNA occupies P site and second tRNA occupies A site. The amino acid of first tRNA and second tRNA are joined by peptide bonds. The initiated tRNA moves to the E site and leaves the complex. The A site tRNA move to the P site and make the A site vacant for the next tRNA. As ribosome move from one end to other, always with the distance of one codon, it is called translocation. Termination. When ribosome arrives at the stop codon, any of the three, UAA, UGA or UAG, कोई भी एक आ गया तो the release factor protein will bind and terminate the translation. Explain processing of hnRNA in eukaryotes with diagram. इसमें सिर्फ डायग्राम भी आता है कभी और कभी सिर्फ शॉर्ट नोट आता है, कभी दोनों भी आ सकता है. So the hnRNA consists of introns and exons. HN for heterogeneous nuclear RNA. It is non-functional. तो जो भी RNA बना है उसको फंक्शनल बनाने के लिए प्रोसेसिंग इज इम्पॉर्टेंट. To become functional mRNA, introns are removed by splicing and exons are joined by the DNA ligase. The exons undergo the process of capping and tailing. In capping, methylated GTP gets added at 5' end, and in tailing, poly A tail gets added at 3' end. This is called polyadenylation. So diagram भी दिया है आपको. Give the name and the functions of enzymes involved in lactose metabolism. So name of the genes given like Z, Y and A. Enzymes, Z बना रहा है बीटा गैलेक्टोसिडेज, Y बनाता है बीटा गैलेक्टोसिडेज परमेज एंड A बनाता है ट्रांस एसिटिलेज. और इन तीनों का फंक्शंस दिया है. इट ब्रेक लैक्टोज into glucose and galactose. It permit lactose to enter inside the cell and it transfer acetyl CoA group to the galactose. Write a note on transcription. It is a process of copying information from DNA to mRNA. The enzyme involved is RNA polymerase. It takes place in the nucleus. There are 3 steps. The promoter is located at 5' end of the structural gene and provide binding site for RNA. The sigma factor help in polymerase to recognize the promoter site. Elongation. The RNA polymerase move along the DNA and unwind it. The RNA nucleotides joined by phosphodiester bond. The mRNA is synthesized in 5' to 3' direction. The RNA polymerase moves in forward direction and add RNA nucleotide complementary to DNA strand sequence until it reaches the terminator sequence. Termination. The terminator sequence is located downstream of the gene. When RNA polymerase reaches terminator, it leaves the DNA and mRNA. Thank you.